So our purpose of identifying the testicles or microanatomy, we are not going to see this big structure. We are going to cut this particular small lobules. That's what we are going to see inside to it. So this particular seminiferous tubules, the inside to it will be our microanatomical focus or point of interest. That's what we are going to talk about. Now, whenever you are going to see these particular tubules or seminiferous tubules, if you cut this particular seminiferous tubules through sections, they may look like circular, oval, or a kind of like a elongated oval shaped structures. Now, within this particular seminiferous tubules, there are two basic types of cells we are going to identify. One is supporting cells, that means they are kind of resident cells. Other is, we call them as migratory cells. Who are migratory cells? They are not inhabitant of that particular region, they are coming from elsewhere. Where from they are coming? They are coming from yolk sac. They travel from the yolk sac and settle down over there. That particular cells which migrates from the yolk sac embryonic region settles down over there, we call them as germ cells. That particular germ cells in future becomes that sperm cells in future as the things progresses or develops with the developmental phase. So whenever you are going to see this particular seminiferous tubules or inside of this particular testicle chambers, we are going to identify two different cells. One is the resident cells that is present within this particular testicles. We call them as the in cells or in-house cells. We call them as this particular supporting cells, and other is the germ cells. The germ cells means the spermatocytes or spermatogonial cells. So these are the two morphology. Other than this, we have some kind of cells which is producing hormones. Those are known as the Leydig cells or interstitial cells. They are present outside the tube, and as we are going to see this microanatomy, they are present accordingly. Now, this is what we are going to study. The layers we talked about, vaginalis, albuginea, vasculosa. Vasculosa is most inside coat of that part, and this is most outside. The vaginalis is basically the peritoneum, which is present or covering to it. Now, then we have this particular albuginea splits into connective septa, which splits into small part, we call them as lobules. And this particular lobules, you cut it and see under microscope, that what we are going to see inside to it, we are going to see the seminiferous tubules. You cut the seminiferous tubules, we are going to see the cells which are present to it. Now, this particular cells which are present in here, we call them as epithelial cells. They are present in the seminiferous tubules, we call them as seminiferous epithelial cells. As we have mentioned, there are two types of cells are there, resident cells and migratory cells. Migratory cells, we call them as germ cells. They means the sperm cells or future sperm cells, and the resident cells, they are present from the beginning of the development. These cells we call them as Sertoli cells. So these resident cells we call them as Sertoli cells, and other cells germ cells, spermatogenic cells, that is going to produce the sperm in future with appropriate environment and appropriate hormonal stimulations is required for it. So these particular regions inside to these seminiferous tubules, the cells which are there, we call them as seminiferous epithelium. This epithelium we call them as complex because they look differently and they are stratified or present in layers. They are having complex layers. The cells do not have a particular shape and size. That's why we call them as complex kind of cells. Now, if you cut section these particular regions, these particular tubes which looks like a circular or oval or elongated circular oval kind of cells, these are cells we call them as seminiferous tubules. Now, inside to this particular seminiferous tubules, these are seminiferous cells or stratified seminiferous cells. Inside to it, we have two kinds of cells present. One is Sertoli cells, which is supporting cells. Second thing is the germ cells, which is the spermatocyte cells. Now, these are present inside to this lumen. So, you see this boundary of the lumen which is demarcated. So, this particular demarcated margin, we see this inside to the lumen. This is another lumen of this particular region. We see the seminiferous tubule. This is another. There are several of them. They are like a heading shoulder to shoulder in this particular region. Now, in between this, we have some space there. Like between this particular seminiferous tubule, we have a tiny space, or we call them a space which is outside the seminiferous tubule. So, these cells we call them as interstitial region. These regions, if you are going to see some kind of cells which are producing hormone, we call them as Leydig interstitial cells. They are present outside the seminiferous tubule. So, now these things becomes clear to us. We are going to see this particular microanatomy of the testis. Now, whenever you are focusing this, we are going to see that seminiferous tubules. They are looking like tubes. The cut sections of the tubes, depending on how you are slashing that, they are taking the shape. Now, inside the tube, we are going to say cells. There are two basic cells are there. One is resident cells, Sertoli cells. Other than non-Sertoli cell, they are germ cells. In case of the sperms and spermatocytes or sperm cell producing cells. Now, how these particular elements are coming? We call this as this is zoom thing, which you are seeing here is more than like 100 zoom microscopes, and we call them as high magnification. You are seeing that. And this is zooming over here is this region, we call them as this particular seminiferous tubules. This is whole seminiferous tubule, this is another, and in between them we have a space. This space is housed by some accumulations of cells, we call them as interstitial cells. These cells produce the hormones, which is the male sex hormone, testosterone, etc., which are produced on these regions. Now, these regions have got two kinds of cell. Inside to it, we have the lumen, which is present in here, and this side away from the lumen, we call them as lumen and adluminal region. So, base of this region is occupied by these cells. We call them as testicular cells, also known as Sertoli cells. So, these are big nuclear cells as you are seeing here, and along with that we say these cells we call them as spermatocytes. Now, these particular spermatocytes, which are germ cells, are present in different forms. That means from the primitive or this old cell to this mature cells as they are moving towards this particular periphery to the central. So, this particular most peripheral region will see that most primitive kind of sperm or germ cells. As we come towards this particular lumen cavity side, we see that more matured versions are coming. Now, quickly we go to different types of sperm cells. Now, we have these sperm cells, we call them as the oldest one, we call them as spermatogonia. They can be classified A, B, and A light stain and B dark stain, like that, and type B. So, these are the oldest versions of the germ cells. They are basically the stem cells from which everything is coming, or great grandfather cells of the spermatocytes. Then we have these particular cells which is evolving from the spermatogonia. We call them as spermatocyte, which is primary one, which gets matured into secondary spermatocytes. Then we get the spermatids, like that. So, this is what we are going to see inside to it, but histologically we cannot distinguish between them. So, what we are going to see, we are understanding this particular different types of the germ cells are there. In case of the male subject, we call them as spermatogonia. We call them as type A, type B. These are most primitive, then which is maturing from the spermatogonia to primary and secondary and finally to the spermatid. These are the maturing cells. These are the germ cells, and these are we call them as testicular cells or old neighbor cells. We call them as Sertoli cells. They are looking like a pyramidal or kind of polyhedral. They have a process which is maybe just like looks like a tree branches moving up and down. Between the Sertoli cells, they have a quick or have a very good tight junction so that nothing escapes between them. So, this particular Sertoli cells, these are present, and along with that we are going to see the outside spermatocyte, outside like seminiferous tubule cell, we call them as interstitial cells. The cells which are present, we call them as Leydig interstitial cells. So, these are the things that we are going to see. So, this particular thing again zoomed over here, this is what we are going to see in this particular basal region. We are going to see these particular cells which we call them as Sertoli cells. Along with that, we have this particular spermatogonia, which is going to be primary spermatocyte, secondary spermatocyte, and most to the lumen side we are going to see the spermatids. That is how they are oriented and arranged. Now, the microstructure of the Sertoli cells, these are like this. So, this is the lumen side, and this is like the side where you see these particular boundaries of this, or away from lumen side. We call them as basal side, where you see this thin basal lamina into which this particular Sertoli cells are placed. So, this whole thing are Sertoli cells, and in between them we have this particular spermatogonial cells placed in here. So, this is away from the lumen side, we call them as adluminal side, and this is towards the lumen side. Now, as we move from this margin to the center, we see that more matured kind of variety of the germ cells are there. Spermatogonial cells are present over this basal region, and we move to the side primary spermatocyte, then secondary, and then finally we get this spermatocyte which is more mature, we call them as spermatids. Now the sperm teats are moving towards the epididymis, stored as spermatozoa, and eventually they get the motility from there and moved out when they are required to. So this is how these particular things are placed. Now this is the flow chart of the things that we should be knowing about how this particular spermatogenesis is going. So coming from this primordial germ cell, going to this particular spermatogonia, type A dark, then it becomes pale, then it becomes type B spermatogonia, then we have this meiosis continuing into it, primary spermatocytes, and it stops at this particular phase, meiosis 1, then it moves to the secondary spermatocyte, sperm teats and spermatozoa finally. This is the whole chart of things. We know this particular thing before that as well from our 10+2 knowledge. I am not just going to spend time on it. This is the process which is going through in the process of this particular sperm cell productions, like how these things are going on, and whole process is known as the spermiogenesis. Now what we are focusing here is this particular microanatomy. So whenever you are going to see this particular schematic pictures, we are going to see identify these rounded structures which is known as the cut sections of the seminiferous tubules. Within the seminiferous tubules, we are going to see two different types of cells: germ cells, which are of different varieties or primary or different forms of the spermatocytes or spermatogonial cells, and as well as the sustentacular cells, which is known as the Sertoli cells. And along with that, in between the space which is lying between the adjacent sperm tubes, we call them as interstitial space, where you are going to find the cells which is present. We call them as Leydig's interstitial cells. They are kind of cells which are producing these hormones which is required for the male testicular maturations. So to summarize these testicles, we are going to see this particular thing. We have the coverings which are made of outside to inside. These are the things. Then we have the spermatotubules which are present in each small lobules, 1 to 3 depending on the spermatoglobules are there. Each of them are occupied by these particular seminiferous tubules, occupied by these particular germ cells and Sertoli cells. Then we have this outside seminiferous tubules, we have the Leydig cells, and these particular Leydig cells are kind of cells which is producing the hormones. Now inside this particular germ cells, we call them as spermatogenic cells. Then we have gonial cells, spermatogonia, spermatocytes, and then we have this particular mature or more mature form, we call them as sperm teats. These are present. So this is what we are going to see histologically whenever we are going to identify. So this is a schematic picture. Now this particular picture is zoomed out here. One single seminiferous tubules that you are seeing here zoomed out along with the cross sections of other two. This is one or half of this you are seeing here. This is magnified one, and this is the lumen side and this is the basal side. So we call them as luminal side or adluminal side. This one is the basal side, which are present in this region, and in between them we have the tiny region, we call them as the interstitial region, where you see these interstitial cells are present to it. So this particular picture is pretty much like easy to identify. The moment you just look through these particular microscopes, these particular regions are pretty much well visible and you can easily identify. There are no chance of mixing up with this particular histology of the spermatocytes or histology of this particular testicles with any other structures. There is way different than the rest of the structures that we have seen so far. Rather easy to identify, or rather very easy. So that's why you put this particular slides more commonly, more frequently during the examinations, which is a kind of like an easy shot to identify. Now, other than this, for the physiology and for anatomical purpose as well, I just want to introduce this particular facts about thing we call them as Blood-Testis Barrier. So this particular thing which is kind of like important for us to understand why this particular barrier is there and what the utility is there. Now for that we just go to this particular picture. Now we see this particular seminiferous tubules which you have cut section on this, and we are going to draw this particular schematic picture again. Now we see that we have the sustentacular cells, also known as Sertoli cells, which are present inside. Now between this sustentacular cells and the spermatogonial cells, which is present a very tight junction which is present. So this particular tight junction between this particular sustentacular cells and this particular spermatogonia existing over here, so that there is no leakage of this particular sperm cells to this particular exterior blood vessels. So it's a kind of barrier which is put through so that this particular exchange of this particular material from the blood system can get into the inside atmosphere of the seminiferous tubules. So why these things are super protected? The reason for this, we do not want to exchange this particular exposure of this protein substances that is present within this particular seminiferous tubules. Now you ask me why this particular things is required or why these things are relevant. Now this particular sperm cells are produced at pubertal age. By that time we have quite exposure to this particular, all this particular required exposure to the protein substances through the immune system. Now by that time the immune training is done. Now after the immune training we have received, the companies have shut. That means this particular immune training process which starts from the lymphocytes, T lymphocytes maturing, B lymphocytes, they are kind of like mature or trained kind of immune system. So that particular exposure is over and those elements which are producing or training this particular our immune system is shut. That means they close the business. And after the closure of business, we are exposing to this particular protein substance which are developed after puberty. So after puberty, if you develop some proteins and that particular protein is exposed to the immune system, that means blood system, that is countered as threats. So we cannot just produce a self-recognizing factors within this particular sperms or spermatogonia or this particular like spermatids like proteins which has developed at the pubertal age. Now if it is exposed to this particular blood system, the body will sense it, this spermatids or this spermatozoa as foreign bodies and result in, if it is exposed, it will be auto-destroyed because of the phagocytosis of the cells which is present. Now if we have this particular destructions of the spermatogonia, spermatids or etc., the species cannot produce through. So that's why we have to keep this particular environment secret so that the blood exposure cannot be put into this particular microenvironment of the sperm production house. So for that we produce this particular barrier system, we call them as Blood-Testis Barrier. So this internal environment of the seminiferous tubules are restricted from the exposure to the blood system. How this is produced? This particular Sertoli cells are producing that. Between this particular Sertoli cells, processes and the cell bodies, they create a tight junction. So nothing escapes between them. In between them, we have this particular things architectured in a way so their exchange of the sperms with this blood system are restricted. And this particular restricted thing is present because of this Blood-Testis Barrier, which is a physical barrier. It's not a physiological thing. This is existing. And this particular thing is produced by these particular Sertoli cells and different parts of the Sertoli cells which is present and taking part in the formations of this. Now, if by any chance, if you lose this particular architecture, so for example, if you have a disease where this particular Sertoli cells are kind of destroyed, maybe having a trauma, maybe having a kind of like infections, or may have some kind of things which is leading to the destructions of the Sertoli cells. By any chance, if you have damage to them, we have a chances of leakage of this particular blood into this system. Once we have this, we have a autoimmune things developed against the sperms. The moment this particular sperms comes out to this particular outside system, this will be destroyed. So that leads to destructions of the sperm cells, which could be a critical medical conditions. So this is why this particular Blood-Testis Barrier is a kind of very important structure that is formed, which is a physical form of isolating the blood system with this particular the seminiferous lumen internal atmospheric structure. In physiology, you will learn about this and you will learn about these diseases which is coming out of these destructions of the Blood-Testis Barriers in pathology, and going to see that in medicine and surgery and the obstetrics gynae, which is one of the leading cause of infertility or male infertility as we progress through this. But coming back to our focus is that we are going to identify this particular seminiferous tubules, and inside to the seminiferous tubules we are going to identify the different kind of cells which is existing, germ cells and non-germ cells, Sertoli cells which are sustentacular cells. This particular sustentacular or Sertoli cells has a number of functions. Besides this particular tight junction formations, this Sertoli cells produce some kind of functions, which is kind of like it develops some kind of proteins which is known as androgen-binding proteins. It creates this blood-testis barrier which is present to it, and along with that it creates this particular environment or gives this nutrition which is required for maturing of this particular different phase of the spermatocytes. These are the prime functions of this particular Sertoli cells. Coming from the Sertoli cells, we have the Leydig cells. The prime functions of the Leydig cells: productions of this testosterone with a pubertal age and synthesis, and this particular pubertal synthesis of this particular testosterone. Once these things testosterone are produced, the Sertoli cell produces a kind of protein substance. We call them as ABP, androgen-binding protein. After this particular androgen-binding protein comes, this particular testosterone is tagged to it, and once this testosterone and androgen-binding protein are tagged to it, we get this particular inside of this particular regions of the seminiferous tubules gets this particular stimulus. So this particular productions of these sperms can be produced through the hormonal stimulations.